Afternoon, folks. Hope everybody is doing well wherever you find yourselves. I am here today to start up some new content with you all to talk a little bit about a thing called cell division. Now, I have placed a handout in the course documents folder and the handouts folder on Blackboard for those of you who are having me for that class. That includes some diagrams of a lot of the things we're going to be talking about here, a lot of the vocabulary. So I strongly recommend that you get yourself that handout and have it available to reference while we are having this discussion. As with all my previous lessons, I'm not going to talk about assignments in here. That will be discussed on the Blackboard site. Now, we've talked about a bunch of different things that cells do. We've talked about how cells manage their energy resources using enzymes. We've talked about how cells move things from one side of the membrane to another with active and passive transport. We've talked about how cells obtain energy through the process of respiration. We have not yet dealt, however, with one very important cellular topic, and that is the topic of how you get new cells. Now think back to your cell theory from way back early on in the course, and you know that one of the final and most significant points of cell theory is that all cells come from the division of existing cells. You do not get new cells from inorganic matter, at least not under the conditions that prevail in the planet that we live on today. In the earliest days of life, we did have to make a transition from inorganic material to cellular matter. There had to be a mechanism by which the first cells emerged. If you take my Bio 2 class, we will talk about that. But for our purposes today, in the world that we live in right now, new cells only come from the division of existing cells. Now, there was a belief that pervaded in biology up until the late 1800s called spontaneous generation, the idea that living things could arise from inorganic sources. For example, it was believed that earthworms would just sort of appear in soil, or that if you had a large enough pile of grain in one place, that mice would spontaneously materialize within it. And when microbiotic organisms were discovered, it was believed for a while that they were emergent from the process of decay, that when food broke down, microorganisms would form within it. Well, that was overturned by a French scientist you might have heard of by the name of Louis Pasteur, P-A-S-T-E-U-R, one of the great biologists of the 19th century. He's the guy who basically invented the modern field of microbiology. The pasteurization process of heating things up just enough to kill the bacteria within them was named after him. And very cogently for our modern situation, he was the guy who first realized that you could expose an organism to a weakened form of a pathogen and cause it to gain immunity so that it would resist the full-strength version of the pathogen when it encountered it later, the thing that we call a vaccine, which we're all hoping for right now, of course. Now, Pasteur did a famous experiment in which he took a quantity of beef broth, put it in a swan-necked flask where dust and particles couldn't easily get in from the outside. Then he heated up the beef broth just hot enough to kill all the bacteria within it, pasteurization as we now call it, and left it. Outside particles could not penetrate the swan-necked flask, and as a result, the beef broth stayed fresh. There were no bacteria in it to make it decay, and it remained that way until Pasteur broke the neck off the flask and made it possible for outside particles to get in. Then and only then did bacteria begin to form within his sample. Pasteur had demonstrated that spontaneous generation didn't work, that it was the bacteria causing the decay process rather than them forming as a product of it. But getting back to the topic at hand, this idea of all cells arise from the division of existing cells. So what we need to talk about now is how this thing called cell division happens. But first, why does it happen? Why do cells divide in the first place? Three big reasons. First off, for growth. If you're a multicellular organism like ourselves, you have a lot of cells in your body. The human body contains upwards of 100 trillion distinct cells. Now, you don't start off with every cell that you're going to have. We all begin as much smaller versions of ourselves, obviously, which means we have to add on new cells. So a multicellular organism engages in cell division for purposes of growth. Even once you're fully matured, growth doesn't necessarily stop. In some species, growth never really stops. Sharks, as long as you keep feeding them, will keep on getting bigger. In some species, certain kinds of growth are continuous, but not all. For example, the teeth of rabbits and rodents will continuously grow until they wear down. That's why it's important if you have a pet rodent to give it something to gnaw on to keep its teeth worn down. And of course, in your body, we have growth that happens after we reach adulthood. We add fatty tissue to our body. We add bone density from working out, and that requires the growth of new cells. Secondly, we engage in cell division for purposes of repair. Not every cell in your body lasts your entire life. You've probably all heard the statement that every 7 years we replace every cell in our body. That's not true, because you've got certain cells in your body that are never replaced. Your neurons, for example, the nerve cells that make up your nerves, your brain, and your spinal cord. Those last your entire life. Same goes for your cardiocytes, the heart muscle cells. But a lot of other kinds of cells in your body, your skin cells, your linings of various ducts and passageways, a lot of cells in our bodies wear out. And a lot of cells can be destroyed or damaged by disease, external injury, toxicity, or whatever. As a result of this, we have to be able to repair the damage. We have to be able to generate new cells to fix damage or replace worn out cells. For example, we have continuous division of cells in the base of our skin to replace skin cells that slowly rise to the surface, die, and flake off. When you exfoliate, you're rubbing off the dead skin cells. Everything you can see on the surface of your skin is dead cells going down about 20 to 40 cell layers. And underneath that, continuously dividing new cells, pushing up to replace the ones that wear off. So, growth and repair. Now, not all organisms are equally good at repairing all things. Some organisms are way more regenerative than we are. If you take a sponge, the very simplest animal of all, cut it up into little pieces and throw them all in a nutrient bath, every piece will grow into a new sponge. Starfish can regenerate entire lost arms. Crabs can regenerate entire lost claws. But if I cut off your hand, you're not going to regenerate that. We're not going to get into the reasons why some species are better at regeneration than others. But suffice to say, in any of those cases, cell division is needed for repair purposes. And finally, reproduction. If all cells come from the division of existing cells, it follows that all living things come from other living things. So the process of reproduction is basically the process of making new life from existing life. And you need cell division to do that. Now, in some species, they're able to reproduce all on their own. There's no partner required. A single individual can make offspring. That is called asexual reproduction. The prefix a means without, so asexual, without sex. Your asexual organisms engage in cell division and set aside a portion of those cells to become a new offspring, which might grow out of their body in some fashion. If you're a single-celled organism, cell division and reproduction are for all intents and purposes the same thing for you. That single cell divides, you get two new cells. We now have two organisms. A lot of single-celled organisms, a lot of prokaryotes and protists, reproduce through a process called binary fission, which is basically cell division for reproductive purposes. Single-celled organism divides into two new single-celled organisms. And finally, reproduction can proceed sexually, in which you have to combine the genetic material of two different individuals. In that case, cell division is needed to make sex cells, better known as gametes, G-A-M-E-T-E. Gametes are the reproductive cells produced by sexually reproducing critters, like ourselves. Your body engages in cell division to make eggs or sperm, which will then combine with other eggs or sperm in order to make new offspring. So we do cell division for growth, we do cell division for reproduction, we do cell division for repair. These are all reasons why this can happen. Now, before we get into the mechanics of how cell division works, I want to talk for a little bit about the life cycle of a cell, the events that a cell goes through from the time that that cell forms through its initial division from another cell until the time that that cell itself ultimately divides. Now, the cell cycle is what we call this. The cell cycle is the sequence of events in the life of a cell between episodes of division. It's 4 distinct phases. We often illustrate this in a circular fashion. You've got a diagram of this in your handout that I described. The 4 steps, basically in order from the time the cell first appears are the G1 phase, the S phase, the G2 phase, and the M phase. Now G1, and the way you write that, it is capital G subscript number 1. The G1 phase, the G stands for gap, because this is a gap between episodes of reproductive activity. During G1, the cell is just doing its normal things. It's carrying out whatever normal activities that cell would perform. But at some point in G1, it is going to hit what we call the G1 checkpoint. At the G1 checkpoint, that cell will either commit to division or not. Why would it not? Sometimes because there are insufficient nutrients available to support the growth of new cells, or other environmental factors that make it a bad thing to be doing at that time. Sometimes it's because that cell quite simply isn't needed for division right now. Sometimes it is because that cell is damaged in some fashion. It's defective in some way, and it's not fit to carry on. Now, at the G1 checkpoint, one of two things will happen. Either the cell will commit to division, and it will go on to the next step, to the S phase, or that cell will leave the cell cycle, and it will go into a separate phase called G0, G superscript number 0. In G0, the cell goes into a quiescent condition, in which it is not preparing for or engaging in division. Sometimes this is temporary, lasts until nutrients are available to foster division or something. Sometimes this is permanent. The cell will go into G0 and never divide again. This is often the case for cells that are incapable of division to begin with. There are a number of types of cells in our body that do not divide at all. We'll explain why that happens a little bit later, but once they form, they are essentially permanently in G0. But let's assume that we have passed the G1 checkpoint. The cell is good, it's cleared for launch, we're ready to go on and start getting ready for division. So we move into the S phase of the cell cycle. S phase stands for synthesis, because during this phase, we are going to synthesize new DNA. We are going to copy the cell's DNA. This is important because both of the new cells we're going to form have to have a set of instructions, right? They need a complete copy of all the genetic information that they are going to need to operate as independent cells. So before we can do anything else, we have to duplicate our genetic information, copy the DNA. So the process of DNA replication happens during the S phase. I should point out that if you are a single-celled organism engaging in binary fission, DNA replication and division happen at the same time, but that's kind of an exception. Now, a word about how this works. When we're talking about the DNA within our cells, if you're a prokaryote, well, your DNA is basically all one unit, right? Remember that circular bacterial chromosome we talked about before? Single unit containing all of your primary genome. Maybe it's got a little plasmid or two floating around it. If a eukaryotic organism is doing this, however, we as eukaryotes have multiple chunks of DNA. Your DNA is not all one unit. Rather, your DNA as a eukaryotic organism is divided into smaller units known as chromosomes. C-H-R-O-M-O-S-O-M-E, chromosomes. Now, chromosomes are basically sections of DNA that are highly compressed to fit lots of DNA into a smaller area. If I took all the DNA out of one of your cells and stretched it out to its full length, every single cell of your body contains about 2 meters of DNA. That's a lot to squeeze into something that's generally less than 100 micrometers in diameter. So to fit it all in, we are going to divide it up into chromosomes, have the DNA in separate chunks, and we are going to compress it down significantly so that more DNA fits into a smaller area. Now, when I'm looking at a chromosome, if you're looking at chromosomes, and you can see a diagram of one in my handout, a chromosome looks like it's got 2 long extensions from a little central circle. That central circle is called the centromere, C-E-N-T-R-O-M-E-R-E. That's the mass of proteins that holds the chromosome together. And then extending from it, you're going to have 2 longer masses of DNA called the arms of the chromosome. Now, if I look at the arm of a chromosome up close, I will find that it is made of loop after loop after loop, weaving back and forth, of a thread-like material called chromatin. C-H-R-O-M-A-T-I-N, chromatin. Chromatin is this kind of thready material. It's packed in very, very tight, weave back and forth, looping back and forth in order to squish as much chromatin into as small an area as possible. Each of the arms of the chromosome is a mass of chromatin. Now let's zoom in closer. Look at that chromatin thread. I will find that the chromatin thread is made out of little bead-like units called nucleosomes. N-U-C-L-E-O-S-O-M-E, nucleosomes. And your chromatin is a long chain of nucleosomes, strung one after another like beads on a necklace. I zoom in on a nucleosome, and I will find that my nucleosome consists of several small spherical packaging proteins called histones. You can think of the histones as being kind of like a spool for thread. And wound around the histones very tightly is the DNA. So we've got this multi-layered packaging system for your DNA to squish as much of it into as little an area as possible. Take the DNA, wrap it up around a histone to form a nucleosome. String together a bunch of nucleosomes, one after the next, and you'll make a long thread of nucleosomes called chromatin. Take the chromatin, weave it back and forth into densely packed loops, and you will form an arm of the chromosome. Attach two arms to a centromere, you got yourself a chromosome. So that's the chromosomal packaging system. That's how we pack that DNA down in. Now, as to how you store this, different species have different numbers of chromosomes. Every species has its own distinct chromosome count, the standard number of chromosomes for the species. We call that the karyotype. K-A-R-Y-O-T-Y-P-E, karyotype. And the word karyotype refers to two different things. One, it refers to both the number and appearance of the chromosomes in a given species. So I could talk about the human karyotype or the dog karyotype or whatever. It also refers to a test that we do to determine that information. So I could say, all right, I did a karyotype on this cell. Now, the human karyotype, anybody know how many chromosomes we've got? Wait, I'm asking you a question you can't answer right now. You're just watching me on video. Humans, the standard human chromotype is to have 46 chromosomes. That is the usual number for our species. All humans, barring chromosomal disorders, are going to have exactly 46 chromosomes. Your DNA is divided up the same way as everyone else's DNA. In other words, you have the same types of chromosomes that everybody else does, and they contain the same genes in the same spots. So my gene for eye color, for example, is located in the same spot on the same chromosome as it is for you. It's a consistent genetic map for our species. When they mapped the human genome, in the Human Genome Project, what they were doing was really locating every gene and its exact position on every chromosome, which is the same for all of us. We number the chromosomes 1 through 23. There are 23 different chromosomes in the human genome. You have 46 chromosomes altogether, but you got 23 different ones, numbered chromosome 1, chromosome 2, and so forth. Reason for that is because you get half of your chromosomes from mom, half of your chromosomes from dad. Your chromosomes are in pairs. You receive 23 from mom, 23 with the same kind of information on them from dad. Those come together to give you your full slate of genetic information. So you have a grand total of 46 chromosomes, 2 copies each of chromosome 1, 2, 3, 4, and so forth. We can look at them, and if I compare your chromosomes to the DNA of your parents, I can even tell you which chromosome specifically came from each of them. You have no chromosomes that are your own, folks. Your chromosomes are all borrowed from mom and dad. So I could do a karyotype on you, and I could karyotype your parents and compare your DNA, and I could say, all right, that chromosome right there, that copy of chromosome 1 in you is the one you got from your mom, and that copy of chromosome 1 is the one you got from your dad. We refer to these often as maternal and paternal homologs. H-O-M-O-L-O-G-O-U-E-S, homologs. When two things are homologous, that means that they are the same or aligned with each other or derived from the same thing. So we talk about homologous pairs of chromosomes, and each homologous pair consists of one paternal chromosome that you got from dad and one maternal chromosome that you got from mom. So whichever of your parents you look more like, you still have equal amounts of genetic information from both of them. So why is it you have mom's nose and dad's eyes? We'll answer that in our question on genetics. But suffice to say that even if you resemble dad more than mom or vice versa, you have actually equal amounts of genetic information in your chromosomes from both parents, 23 chromosomes from each. Now, that's the human karyotype. We have 46 chromosomes in 23 pairs. Other species have different numbers. Fruit flies only have 2. Dogs have 48. Oak trees have more than 1,000. Humans are nowhere near the most complex organisms, chromosomally speaking. Lots of plants and animals have way more genetic information than you do. Now, the standard for most prokaryotic life is to have 2 copies of each chromosome, to inherit 2 copies, one from mom, one from dad. Nearly every cell in your body, what we call the somatic cells, the term somatic, S-O-M-A-T-I-C, somatic. Somatic cells refer to normal body cells. So skin cells, bone cells, muscle cells, those are all somatic cells. Every cell that isn't a sex cell is a somatic cell. And the normal condition for most eukaryotes is to have paired chromosomes, 2 copies of each chromosome in all of their somatic cells. One maternal, one paternal. We call this condition being a diploid cell. D-I-P-L-O-I-D. If I say that a cell is diploid, then that means it has paired chromosomes, 2 copies of each. We can also describe a diploid cell as a 2N cell. That's a common usage, number 2, lowercase letter n. So the normal condition for most animal cells, most plant cells, and so forth is to be diploid, to have 2 copies of each chromosome. In plants, this sometimes varies. A lot of plants have larger numbers of chromosomes, more copies of each one. It is not unusual for plants to have 4 or 8 or even 12 copies of a chromosome. We didn't get to do the strawberry lab in class, at least not as yet, but strawberries are octoploid. Octo for 8. Strawberries have 8 copies of each chromosome. But we're going to think in terms of diploid, because that is the normal condition for most animals, humans included. Getting back to this, kind of winding back to S phase, we kind of had to go through and talk a little bit about chromosomes there first. So during the S phase of the cell cycle, you're going to copy all your DNA, which means that you are temporarily going to duplicate all of your chromosomes. You're going to make copies of every chromosome that you have. That way, all of the new cells end up with a complete copy of the genetic information. So in humans, that means we temporarily go from having 46 chromosomes, 2 of each, to having 92 chromosomes, 4 of each. The condition of having 4 copies of each chromosome is to be tetraploid, T-E-T-R-A, tetra. That prefix means 4, like remember the old game Tetris, with the little groups of 4 cubes. But so if a cell is tetraploid, it has 4 copies of each chromosome. During S phase, your cells temporarily become tetraploid and have 92 chromosomes, 4 of each, so that way you can pass on a complete set of 46 to each of your offspring. Or to each of your new cells, I should say. Offspring, that's a different story. We'll get to that shortly. So S phase, we duplicate our chromosomes. The cell temporarily becomes a tetraploid cell. When the chromosomes are duplicated, incidentally, what's going to happen is that each chromatid, the chromatids are the masses of chromosome extending from the centromere, each chromatid is going to be copied, and you're going to have, instead of like a sort of propeller-looking thing, like one centromere with 2 arms sticking out of it, you're going to go to having a sort of X-shaped thing, where you've got a centromere with 4 chromatids sticking out of it. Both of the new chromosomes that you just formed share a common centromere, at least at first. The term that is often used for them at that time is we call them sister chromatids, whenever they're joined together like that, and you have 2 copies of each arm on each chromosome. All right? Sound good? So that's S phase. We have copied our genetic information. We have doubled our chromosome counts. Let's move on to a higher stage here. Now that we finished S phase, we've copied our genetic information, we move on to G2, G subscript number 2, gap 2, which is when we are actively preparing for cell division. In G2, the cell is dedicating most of its energy and resources to getting ready to divide. We don't just duplicate the chromosomes, folks. We have to duplicate everything. We have to duplicate all the organelles. We have to increase the size of the cell so it can split into 2 new functional cells. We have to increase the volume of cytoplasm. So during G2, the cell is enlarging, duplicating its internal structures, duplicating everything other than the DNA in preparation for division. This includes ordering its mitochondria to go through their own cycle of division. The mitochondria have their own DNA. They go through a division process completely separate from that of the rest of the cell. But they're doing it at the same time as G2 is happening. So G2 is a pretty energy-intensive stage. This is when the cell is devoting most of its resources to prep. Now, at the end of G2, we are going to hit the G2 checkpoint. And in the G2 checkpoint, basically, we're going to check the cell over and see if everything is kosher, if the cell has all the parts that it needs, if its DNA is intact and ready to go, if this cell is in fact fit for division. If the cell passes the G2 checkpoint, it goes on to the next stage. It goes on to M phase. If the cell fails the G2 checkpoint, one of 2 things will happen. One, the cell will attempt repair. We will try to repair the cell and get it fit for division. If it can be repaired, awesome. But if it cannot be repaired, particularly if the DNA itself is damaged, if the DNA is damaged in some fashion due to a mutation event, if the DNA did not copy properly during the S phase, if the DNA isn't fit, if the cell is not able to be a good fit cell that should divide, it will fail the G2 checkpoint, at which time it will leave the cell cycle. And a cell that fails G2 will typically undergo apoptosis, A-P-O-P-T-O-S-I-S, apoptosis, sometimes referred to as programmed cell death. Apoptosis is effectively cellular suicide. It's a self-destruct command built into every cell of your body that basically says, hey, I've been compromised, shut me down. And if the cell fails the G2 checkpoint, typically that cell will go into apoptosis. It will self-destruct. It will digest itself from within. This is a vital mechanism, incidentally, for stopping cancer cells. Our body shuts down most cancers via apoptosis before you ever know you have them. And fundamentally, cancer is a failure of apoptosis. But we'll talk about that in a future chapter. So the cell either passes the G2 checkpoint, goes into M phase, or fails the G2 checkpoint, in which case it typically undergoes apoptosis. By the way, fun little mythological story for you here. We get a lot of our terminology in the biosciences from Greek, but the term apoptosis comes from ancient Egyptian. In Egyptian mythology, Apep, A-P-E-P, is the serpent that eats its own tail and surrounds the world and will devour the world at the end of time. So the term apoptosis, the cell devouring itself, comes from the root name apop or apep from Egyptian mythology. It's the only word in biology that comes from ancient Egyptian. All right, so we finish apoptosis. No, we don't finish apoptosis. We're not going to go anywhere if we go through apoptosis. Let's say that our cell has passed the G2 checkpoint. And now we are moving on to the M phase. The M phase, or mitotic phase, is when the cell is going to go through a cycle of division. 2 key events happen during M phase. One of them is going to be either mitosis or meiosis, the 2 distinct pathways to cell division. We'll talk about the difference between them in a moment. Mitosis, M-I-T-O-S-I-S. Meiosis, M-E-I-O-S-I-S. Mitosis or meiosis. Any given cell will go through one or the other of those processes during M phase. Following mitosis or meiosis, the cell will then go through cytokinesis, C-Y-T-O-K-I-N-E-S-I-S, cytokinesis. Cyto means cell, kinesis from kinetic, like motion. So it basically says the cell moves. What's happening here is the cell is stretching out, splitting up its cytoplasm and its other contents, and separating into two new daughter cells. Oh, terminology I didn't give you yet. When we're talking about cell division, the original cell that we start with is the mother cell. The new cells that we get at the end of division are the daughter cells. So we start with the mother cell, we end up with daughter cells. I should point out, it's really important to know here, that when you're done, you don't have one original and one copy. The mother cell is destroyed during the process of division. It is basically torn in half, and its contents split up among two new cells. So when all is said and done, what you're going to have is going to be two totally new cells, two daughter cells. Neither of them is the original mother cell. Both contain parts of the original mother cell. Now, a lot of people confuse the terms mitosis or meiosis with actual cell division. In reality, mitosis and meiosis are not cell division. They are the preparatory stage necessary for cell division. What's ultimately happening in mitosis or meiosis is that we are sorting and splitting up the chromosomes. That's what those two processes are for. They are for sorting and splitting up the chromosomes. Cytokinesis is the actual division when all this happens. Cytokinesis is when the cell actually splits in two. And following that, following cytokinesis, we go back into G1. Each of those novel cells enters its own G1 phase, and the cell cycle starts over again. How long does this take? Really depends on the organism, depends on what kind of cell. In bacteria, under optimal conditions, the entire cycle might take 20 minutes from the time that the cell forms till the time the bacterium splits into two new bacteria. In your body, once a cell commits to division, it will typically undergo a mitotic process that lasts about 24 hours from the G1 checkpoint till the end of M phase. Mitosis itself is a lot quicker. In human cells, mitosis normally takes 2 to 2.5 hours, which means that basically we only spend about 10% of that cell cycle from G1 checkpoint to division in mitosis or meiosis. So that is our cell cycle. G1, gap 1, normal life stage. G1 checkpoint, we commit to division or we leave the cell cycle and go into G0. S phase, duplicate our chromosomes, synthesize new DNA, temporarily double the chromosome counts. In most cells, that means becoming tetraploid rather than diploid. G2, gap 2 phase, active preparation for division. Cell grows, enlarges, duplicates its other contents. If that cell has mitochondria or chloroplasts, they go through their own cycle of division at this time. And finally, M phase, which features either mitosis or meiosis, depending on what kind of cell we're dealing with, and then ends with cytokinesis, in which the cell actually splits into two new daughter cells. And that's our cycle. All right, feeling good? Okay, so how is this done? Now, the lead up to M phase, G1, S, and G2, collectively that is called interphase, I-N-T-E-R-P-H-A-S-E. And in a lot of textbooks describing the steps of mitosis, they will list interphase at the beginning. But interphase is not part of mitosis. Interphase is all the parts of the cell cycle that happen before M phase. So G1, S, G2, they are not part of mitosis. They are pre-mitotic events. Likewise, cytokinesis is often listed, if you're looking at the steps of mitosis or meiosis, at the very end of the process. Cytokinesis is not part of mitosis or meiosis. It is a separate event that happens after mitosis or meiosis. M phase has two things in it, either your mitosis or meiosis, and then cytokinesis. So that's a good listing of the sequence of events: interphase, mitosis or meiosis, cytokinesis, but don't lump them all together and say all of that is mitosis. Now, my actual mitosis or meiosis, like I said, the purpose of them fundamentally is to divide up the chromosomes, to split up the genetic information between two cells. or prep basically for dividing up the chromosomes so that cytokinesis can do that. Now, what's the difference? Mitosis is the older of the two processes. It was the first cell division process to evolve. It's very similar to binary fission. In mitosis, you were going to make identical copies of the mother cell. You start off with a diploid mother cell. In S phase, it doubles its chromosome count, becomes tetraploid, undergoes the process of mitosis, after which it splits into two new daughter cells, both of which are diploid, just like the mother cell. And if everything went well, both of which are genetically identical to the mother cell. I say everything went well because mutation is a thing that can happen along the way. We'll talk about mutation in a future chapter. Mitosis is good for repair if you're growing new cells to fix damage or worn out cells. Mitosis is good for growth if I'm adding cells to my body. Mitosis is good for reproduction if I am an asexual organism and I am setting aside portions of my cells to become new offspring, or if I am a single-celled organism undergoing binary fission. I'm basically undergoing mitosis. And mitosis is perfectly great if what I want are identical diploid copies of the mother cell. We don't always want that. You are a sexually reproducing organism. In other words, for you to reproduce, just like every animal, we have to combine, well, there are a couple animals that are asexual. Long story, we'll get to that later. Remember, every rule in biology has exceptions, including the rule that every rule has exceptions. But if you're a sexually reproducing animal like ourselves, you cannot make offspring all by yourself. You have to combine your genetic information with somebody else's genetic information. Now, here's the thing. I want to keep the karyotype consistent. I want every human to have 46 chromosomes. Having even one extra chromosome is a problem, as noted by things like Down syndrome. Having too few chromosomes is a problem. Most organisms with too few chromosomes don't survive at all. So we want to make the chromosome count consistent from one generation to the next. We want to make sure that you have 46 chromosomes and your kids do as well. But here's the thing. For that to happen, you can't pass on every chromosome to your kids. If you and your partner both passed on all your chromosomes, the kid would have 92 chromosomes, and their kids would have 184 chromosomes. We would double the chromosome count every single generation. Not going to work. So what needs to happen here is a way for you to only pass on half of your chromosomes, 23 rather than 46 for humans. That way, your 23 combined with your partner's 23, you end up with an offspring that has 46 chromosomes in pairs, the way things are supposed to be. So we need a slightly different division process for this, and that is where meiosis comes in. The purpose of meiosis is not to make identical copies of the mother cell. The purpose of meiosis is to make non-identical sex cells, which are called gametes. There are two kinds of gametes. You have oocytes, which is spelled O-O-C-Y-T-E, oocyte. You pronounce both O's separately. It's not oocytes. The oocyte is the female gametes, what we often refer to as the egg. It is the larger and more complex of the two because it has to do the more complex job of developing into the full offspring. The male gamete is called the spermatozoa, S-P-E-R-M-A-T-O-Z-O-A, spermatozoa, often just called sperm for short. Sperm are much smaller and much less sophisticated than eggs because a sperm only has one job, and that is to deliver dad's chromosomes to the egg. The sperm is a vehicle to carry 23 chromosomes to the egg cell, fuses with the egg, and then from that point on, you have a single cell with 46 chromosomes, a complete set of genetic information, half mom, half dad. That fertilized egg, by the way, is called a zygote, Z-Y-G-O-T-E. Great Scrabble word for you there. Now, the zygote, once it's formed, is going to go through its own cycle of division and start going through mitosis to make more cells that will eventually grow into you. We're going to talk about that side of things a little further down the line. But right now, the important thing to know is that sexually reproducing organisms get 23 chromosomes from the egg, or oocyte, 23 chromosomes from the sperm or spermatozoa, at least humans. Other species, chromosome count will be different. So, to make gametes, we have to go through a slightly different process. Mitosis is great at making identical copies of the mother cell that are diploid. Meiosis is going to happen a bit differently. In meiosis, we are going to undergo 2 cycles of division. There are 2 rounds of meiosis, called meiosis 1 and meiosis 2. Use Roman numerals to designate those. In meiosis 1, we're going to take that initial mother cell, double its chromosome count in S phase before this all begins. So we start off meiosis 1 with a tetraploid cell. And in meiosis 1, it's going to split into 2 diploid daughter cells. But we're not done. Each of those diploid daughter cells will then undergo meiosis 2. And in meiosis 2, they will each split again. And from each diploid daughter cell, you will get a haploid or 1N gametes. Gametes are haploid. Think haploid half. They have half the normal chromosome counts. So each of those gametes has only 23 chromosomes. Their chromosomes are unpaired. They have 1 out of each pair. So from one mother cell, you started off mitosis with a diploid 2N mother cell. During the S phase, meiosis, excuse me. During the S phase, you doubled its chromosome counts to make a tetraploid or 4N cell. Meiosis 1 split that tetraploid cell into 2 new diploid cells, and then in meiosis 2, each of them splits again to form a grand total of 4 haploid gametes. 4 eggs or 4 sperm from the original mother cell. Along the way, several different things are going to happen in meiosis that make it differ from mitosis in order to cause them to come out as genetically distinct cells. You are going to find there are lots of similarities between the two. A lot of the things I'm going to tell you about in meiosis and mitosis share similar names, similar events happen. Meiosis evolved from mitosis. It was a branching off from that basic process of primitive cell division with the first living things when sexual reproduction first evolved about 2 billion years ago. Now, nearly every cell is going to undergo mitosis eventually. Most cells will undergo a process of mitosis at some point in their existence. As I said before, there are a few types of non-mitotic cells. We'll talk about them at the end and find out why they don't divide. So most cells undergo mitosis. Most cells will never undergo meiosis. Meiosis is not needed for most cells. For starters, meiosis only happens in sexually reproducing organisms. Asexual organisms don't need it because they don't need gametes. They don't need eggs or sperm. They reproduce all on their own. 2, not every cell of your body is involved in making eggs or sperm. So most of your cells also never undergo meiosis. In fact, the only cells that ever undergo meiosis are a population of cells called the germline cells, sometimes just called germ cells, G-E-R-M. Germline cells are a special population of cells found in the organs called the gonads, G-O-N-A-D, gonads. Gonads are the reproductive organs. The female gonad is called the ovary, O-V-A-R-Y, or plural ovaries, I-E-S instead of Y. And most female organisms have 2 of them. The male gonads are called the testes, T-E-S-T. ES is the plural form. If you're talking about just one, the proper form is testis, T-E-S-T-I-S. So 1 testis or 2 testes, and testes is ES, not IES. And normally we're going to use the plural form because, like ovaries, testes normally come in pairs. These are the organs where gametes are made, and the germline cells within them are the only cells that ever undergo meiosis. Fun bit of trivia for you about this, by the way. In evolutionary terms, we say that men are the quote-unquote fast sex, or I should say male organisms, and female organisms are the quote-unquote slow sex. What I mean by this is that male organisms produce lots of gametes very fast. Male organisms make gametes extremely quickly. They make little tiny small cells all the time. From the time a male organism hits puberty, it will generally continue to generate gametes on a constant basis for the rest of its life. although they'll slow down as they get older. A healthy adult male human can make approximately 5 billion with a B, sperm every single week. The female of the species is considered the slow sex. Eggs are much slower to develop. They're larger and more complex cells. They have to do a more sophisticated job. Sperm just has to get to the egg and deliver its chromosomes. Egg then has to implant into the wall of the uterus and develop into a new offspring. So the egg is a bigger, more sophisticated cell, requires more energy investment by mom to make. In fact, most female organisms start making eggs before they are even born. There used to be a belief that you had all the eggs you were ever going to have by the time you were born. We now know that is not entirely correct. You continue making eggs, although at a slower level, throughout your adult life until you hit menopause. Normally, we're going to find, therefore, the egg is a higher investment thing. Female organisms can't reproduce as fast as male organisms because once the female is fertilized, she basically has to finish cooking that offspring before she can be fertilized again. On the other hand, male organisms can fertilize as many females as they can inseminate. This is why sperm are produced at such higher numbers, because the male reproductive strategy from an evolutionary level is fertilize lots of offspring. The female reproductive strategy is invest lots of energy in smaller numbers of offspring. And that has a lot of implications for how sexual behavior adapts in different species. Take my Bio 2 class, we'll talk about that in great detail. A thing called sexual selection. It's really fascinating. Also, you need a lot of sperm because most of them are never going to get anywhere close to the egg. Whenever the average male adult human ejaculates, he releases somewhere between 300 and 500 million sperm. And most of them will die before they ever get close to the egg. The internal environment of the vagina and the uterus is actively hostile to sperm. It has all kinds of defenses against invading external cells, against bacteria and other things that could potentially get in through the vaginal orifice. All those defenses that protect the female body against bacteria coming in by way of the vagina also attack the sperm. So most of the sperm are going to die by the defenses of the normal environment of the female reproductive tract. Furthermore, sperm are dumb. They're dudes. They don't ask for directions, right? A lot of them are going to swim around, get lost along the way, bump into the walls of the uterus, try to fertilize things that aren't eggs. Most of them are not going to ever get anywhere close to the egg. So releasing 300 million of them at once means that maybe 1 or 2 of them will have a shot of making it all the way to the egg. Make sense? Okay. So this is why males are the fast sex. This is why females are the quote-unquote slow sex. All right. So that is our backdrop to all of this. We now know about how chromosomes package DNA. We now know about the cell cycle. We know some reasons why cell division would take place. And we know the basic differences between the 2 major types of cell division, mitosis and meiosis. Of course, we are not done. We have some much more complex discussion to have about how these processes actually unfold. But I think I have given you a pretty solid lesson for the time being. So we're going to tie this off. I have talked to you now about the fundamentals of cell division. In my next lesson, we're going to go into the details of how mitosis and meiosis actually work. Hold on to that handout. You're going to want those diagrams handy for your reference. Hope you're all having a great day. I'll talk to you later. Good night.